Remember that there are two very different processes that make ATP from ADP plus a phosphate group in microbial cells and other kinds of cells. There's this process called oxidative phosphorylation, where the phosphate group is being added on to the ADP molecule to make ATP using that really critical enzyme called ATP synthase found in the cell membrane. Remember, oxidative phosphorylation occurs after cellular respiration, and it's very efficient. For each mole of simple sugar, you get between 30 and about 36 molecules of ATP made. Very efficient. The other mechanism is substrate level phosphorylation, and we talked about this in the last lecture series. Substrate level phosphorylation is a process that makes ATP, but this one occurs during cellular respiration. Specifically in the first part of respiration, what we think of as glycolysis, and in the part of cellular respiration called the Krebs cycle. Two times when a small amount of ATP is being made. This is not a particularly efficient way to make ATP. You get a relatively few molecules of ATP for each molecule of glucose. Substrate level phosphorylation occurs during cellular respiration in a couple of different places. It also occurs during fermentation. And remember, that's because fermentation begins with this process of glycolysis or a cousin of glycolysis, depending on which simple sugar is being used. If glucose is the simple sugar, we call its breakdown, its oxidation, glycolysis. But if a microbe uses a different simple sugar, you have a different variation on glycolysis. The same thing is happening though, whether you're doing glycolysis or one of its sort of cousin reactions. You are making a little bit of ATP along the way using this process called substrate level phosphorylation. So there are different ways that ATP gets made in a cell. Some microbes make it doing cellular respiration and oxidative phosphorylation. And in that group of microbes, some of them are aerobic. Some of them use oxygen as their final electron acceptor molecule. And then there are anaerobic microbes who use nitrate or sulfate or carbonate or some other molecule as their final electron acceptor molecule. Some microbes do cellular respiration and oxidative phosphorylation, or fermentation and substrate level phosphorylation. They can do both of them. Remember, our facultative anaerobes, that was the group we said most microbes fall into. Facultative anaerobes. Those are microbes that prefer to do cellular respiration and oxidative phosphorylation. They prefer to use oxygen if it's available. But if there is no oxygen available, they just do another process, like fermentation and substrate level phosphorylation. Doesn't matter to them, they can do both of them. Then there's other microbes that don't do oxidative phosphorylation at all. They're going to be doing fermentation exclusively. So those pure fermenters, as we call them, are not doing oxidative phosphorylation. They're doing substrate level phosphorylation to make their ATP. Remember, one of the most interesting things about microbial metabolism is that there is incredible diversity. You and I, as animals, are only able to do cellular respiration and oxidative phosphorylation to do our ATP production every day. We can ferment temporarily. We can ferment for very short periods of time. But if we don't have oxygen available, we die. There are a lot of microbes out there that can sort of pick and choose among these metabolic processes. Um, they are much more diverse than we are in their metabolic capabilities. Next, I want to get into the specific or the more specific details of cellular respiration and oxidative phosphorylation. So here's where we're going to stop talking so generally about this long series of reactions, and we're going to start discussing the specific details that we need to know as students of microbiology. Let's begin by talking about this process called glycolysis. If a microbe is going to use glucose as a simple sugar to do cellular respiration, it's going to break that glucose down, beginning with a series of reactions that we call glycolysis. There are versions of glycolysis, though. There are versions of this series of reactions, and you've probably heard some other names for glycolysis. Maybe you've heard of the Embden-Meyerhof pathway, maybe not. But if you have, that's one of those versions of glycolysis that is used by microbes that are using different sugars for this process. So, glycolysis, or its equivalent, its cousin mechanism, is a series of about 10 reactions. It's a long pathway. It's not simple at all. But it breaks that sugar down into the first metabolite, if you will, and that's called pyruvate. We talked about this pyruvate molecule as being critical in fermentation. Well, once the cell creates pyruvate from the simple sugar, it doesn't stop there. Remember, the microbe is going to take that simple sugar and break it down or oxidize it all the way to carbon dioxide. Glycolysis is just the beginning. Glycolysis takes the 6-carbon glucose, for example, and creates 2 3-carbon molecules of pyruvate. Then the pyruvate goes into a second metabolic reaction called pyruvate processing. What happens in pyruvate processing is the pyruvate gets broken down a little more into a molecule called acetyl-CoA, and we'll look at that in just a moment. The acetyl-CoA then goes into a third series of reactions called the Krebs cycle, or again, an equivalent pathway. All of the electrons in all of the covalent bonds in the glucose or the other simple sugar by the end of the Krebs cycle have been released. We're left, in other words, at the end of the Krebs cycle with carbon dioxide. Now, the next part of the process, the first part of oxidative phosphorylation, is this structure called the electron transport chain. All the electron transport chain is a group of protein. They're actually proteins that are clustered together, and there's a whole series of them sitting in the membrane, the cell membrane of a microbial cell. And it's their job to, again, harvest energy from all of the electrons that have been released from the sugar. So the electrons get passed through this collection of proteins, and their energy is harvested. Remember, the next thing that happens at the bottom of that electron transport chain is some molecule has to be there to accept those electrons, to take them in. Some molecule has to get reduced at the end of that electron transport chain. Meanwhile, all of the energy that was harvested is going to be used to build the proton gradient. The energy is going to fuel a pump protein that will start pumping hydrogen ions from the cytoplasm into the periplasmic space. and the cell builds this gradient, this difference in concentration of hydrogen ions. That gradient is then allowed to resolve. It's allowed to go away by having the hydrogen ions flow back into the cytoplasm through the molecule called ATP synthase. They are literally, these hydrogen ions, going to flow through a channel through the membrane, a tunnel through the membrane that is part of this ATP synthase molecule. And as they physically flow, as they physically pass through that tunnel, that motion, that movement, that kinetic energy is going to cause the rest of the ATP synthase molecule to physically spin. The rest of that ATP synthase molecule hangs in the cytoplasm. I'll show you a picture in a minute. And it physically rotates as the hydrogen ions flow through the tunnel. And it's that motion, that kinetic energy, that then gets transformed or converted into chemical bonds. And the bonds are between a phosphate group and the ADP molecule, which makes ATP. Glycolysis and pyruvate processing and the Krebs cycle take that glucose or other simple sugar all the way down to a one carbon molecule, carbon dioxide. Glycolysis and pyruvate processing and the Krebs cycle are a long series of oxidation reduction reactions. Glucose gets oxidized to carbon dioxide. Remember, lose electrons, oxidation. And as the glycolysis is occurring, as those electrons get released, they don't go directly to the electron transport chain. Instead, there are these little helper molecules, if you will, also called electron carriers, that are going to get reduced by gaining the electrons that were released from the sugar. These little molecules, and you may have heard of these are things like NADH and FADH2. These are little molecules that just happen to have a high electronegativity, and they're able to accept electrons very readily. They get reduced as the sugar gets oxidized. Those electron carrier molecules are then going to get oxidized when they lose those electrons to the electron transport chain. The electron carrier gets oxidized. The electron transport chain gets reduced. Then those electrons pass down the electron transport chain. Every time the electrons go from protein to protein, the protein that is losing the electron is oxidized. The protein that gains the electron gets reduced. It's just oxidation reduction, oxidation reduction, oxidation reduction. At the end of the electron transport chain, the final protein in that chain gets oxidized. It loses the electrons. And the final electron acceptor molecule, the oxygen, the nitrate, the sulfate, whatever, gets reduced. It accepts those electrons for the last time. That's why we refer to it as the final electron acceptor, and it gets reduced. So, oxidation, reduction, oxidation, reduction, oxidation, reduction. And we've taken a sugar and we have set free all of the energy in that molecule. So the electron transport chain is sitting in the cell membrane of a microbial cell. And again, it's just these complexes of protein. It acts like a staircase, as I was saying, in that you have to think about the electrons sort of coming into the electron transport chain the way you come into the top of a staircase. And as the electrons get passed from one protein to the next, to the next, to the next, it's like they're walking down that staircase. And as they take each step in the staircase, they lose a little of their energy. The electron transport chain harvests it, takes it from the electron. It's important to remember that the electron transport chain is harvesting energy from these electrons. The electrons are traveling down the electron transport chain, and the chain is taking their energy away from them. That energy will be used to pump the protons from the cytoplasm to the periplasmic space and create our proton gradient. And then those protons will flow through ATP synthase and generate ATP. On this slide, we can see an artist's drawing of the ATP synthase molecule. If you look on the left, you can see a drawing of a piece of membrane. So this represents the cell membrane of a microbial cell. Remember, the circles are heads and the squiggly lines are tails. So we have two layers in a membrane. We have a bilayer. The heads are polar. So heads face the outside of the cell, heads face the inside of the cell. The tails are nonpolar, so they sort of cluster together with van der Waals interactions. Here's our cytoplasm in here. And look, here is our enormous enzyme called ATP synthase. Look at this thing. People have sort of deconstructed the shape of this protein and discovered that part of the molecule sits embedded in the cell membrane. And it's shaped in a way so that it literally has a tunnel through it. It literally has an opening or a channel right through that portion of the molecule. And the hydrogen ions that they've drawn here, they're going to flow right through that tunnel into the cytoplasm. And as they do, they're going to cause this portion of the molecule, what looks like a pumpkin, they're going to cause this portion to spin, to rotate, to literally turn around and around and around. It's really quite remarkable. It's that motion, that kinetic energy, that does this, that allows this phosphate group to be attached to this ADP molecule and create ATP.